Welcome to your lesson on DNA replication and repair. We have to start off by taking you back to the good old days of grade 10 when life was simpler and you learned for the first time about the actual cell cycle. So I just want to jog your memory. I want to remind you that cells are normally found in G1. That's when they're going through growth. They're doing their normal metabolic activity. So anything to do with producing energy, that's what's going on. They're making their proteins. They're living their happy little lives. They're do whatever they need to do as a normal cell. So if they are a cell that produces, let's say, human insulin, it's a pancreas cell, then that's what it's doing. It's doing all of its normal functions during that particular time of life. Then it gets a signal that says it's time to divide, and it goes through the S phase. The only thing really that happens here in S phase is that the DNA gets duplicated, and that's really what we're focusing on in this lesson today. Then, after that, you start to prepare for cell division. And then you actually go through this little slice here, which would be the actual mitosis itself. And of course, cytokinesis. And then the two cells are, the brand new cells are now going to go back into G1 and live their normal life until they get that signal again. So what we're dealing with in this lesson with DNA replication is everything that's happening during S phase. Okay, so during the duplication. So 1958. You have Matthew Meselson and Franklin Stahl, and they're experimenting with Escherichia coli bacteria. You've heard of Escherichia before, but we've always called it just E. coli. Normally what they do with these binary names is they'll take that first letter and they'll just say this is the entire genus, the whole genus is described as E, and then this is the version of Escherichia that we're talking about. So this is its more specific name, or what we call a specific epithet. So genus name, species name. So this is the coli version of Escherichia. The reason it's called coli is because it lives in your colon. It also lives in soil areas, and you can find a lot of E. coli on like feces and whatnot that are in the soil. So just be aware, and that's where E. coli lives. Now what they were talking about here in 1958. Now keep in mind, 1953 we discovered the structure of DNA with confirmed with the calculations made by Watson and Crick. And so it's only 5 years later, so we're actually doing a massive amount of study in between 1953 and 1958. In only 5 years, we're already getting to the point where we're trying to decide how, now that we know the structure, how is DNA actually being used? And so here's the first thought that they had. They thought there's two ways that DNA could be reproduced. If the replication was semi-conservative, that means that here's your original strand here. Now they know that there's hydrogen bonds in between. They thought it could be that this little midsection here just unzips, and you can see there's the two halves, the original halves on either side here. And then you just get opposite complementary strands for both of those guys, and then you're done. And then you've got two brand new strands, and you have saved half of the original strand for each new version. So that's called semi-conservative replication. They said it could be done this way, or maybe it's more like this, where you take the entire copy, the entire original, and it stays together, but somehow you make a complete replicate of the first one, so that you would see, here's our original, it stays together, it's fully conserved, we've saved the entire helix, conservative, and then the new strand is a duplication of both of those mini strands. Okay, so what they did is they tried to prove which way it could be, and what they were doing was they were feeding bacteria on a medium. Now, in this case, medium doesn't mean middle-sized, it means food, what kind of food you're feeding them. So they fed bacteria food that happened to have a heavy version of nitrogen. Nitrogen is normally N14 for its atomic mass, and this has one extra neutron, which allows it to be a little bit heavier, but it literally is heavier. A bacteria that has been eating N15 for all of its life will be literally heavier than an identical bacteria that's been eating N14. You can actually weigh the difference. So they feed these guys for 17 generations. So, grandma, great-grandma, great-great-great-great-great-great-great-great-great, however many it takes to make 17 generations. Basically what they've done is that after 17 generations, they're pretty sure that every bacteria that exists, every single bacteria must have heavy nitrogen in their DNA. Okay? now that they're all heavy, that's great. The very next thing that they do is, after 17 generations, they move that heavy bacteria into a food or a medium that only has the normal light nitrogen. And then the very next generation that is born, the only food that it's available to make its DNA only has light nitrogen available. So that's going to mean that any successive generations after this point will have to use light nitrogen to make the new DNA, which means they will literally have lighter DNA. Then what they did, Meselson and Stahl, compared the DNA densities after the switch, and then for the next few generations. Now, if the results were conservative, if we took that original strand and kept the helix together, so then we would have had a heavy helix in the original parents, then you would have had heavy next generation and a light next generation. The lights would have continued to make lights. The heavy would have continued to split into light and heavy generations. And here's what you would have seen. You would have taken the DNA and you would have thrown it into a machine called the centrifuge. These are just little tiny vials that we use. You pop the DNA into this liquid here. It's some kind of a salt solution just to keep it safe. You throw it into the microfuge. The microfuge spins it as fast as possible. And what will happen is, just like a washer on the end of a string, when you start to spin it, the heavy stuff moves to the outside of the centrifuge and all the lightest stuff stays to the middle. So we would have all the light stuff towards the cap, which is in the middle of the centrifuge, and then the bottoms is sticking out of the centrifuge, so all the heavy stuff would migrate. So we're able to separate things determined by their density. Okay, so light strands would appear close to the cap, intermediate would be in the middle, heavy would be at the bottom. So if this is what was happening, if the results were conservative, they would expect that after the initial parents were tested, before we even threw them into the light nitrogen, we would expect that all the parents in that generation would all be heavy, have heavy DNA. Then, after one generation of doubling, all of the brand new DNA, you still have the original heavy, but you should also see bands of light DNA showing up because the new babies will have light DNA. Then, over successive generations, after another 12 hours, because it only takes 12 hours for E. coli to double up, after another set of generations, you should see more and more and more light nitrogen bands versus less and less heavy. And that's what you would see here. See, you got 3 times as many light DNA strands as you do heavy. Okay, so you'd expect this: heavy, then a light and a heavy, and then more and more light and less heavy. Now, if the results were semi-conservative, then you would expect that each of these strands would separate and each one would be used as a template for the new strand. Well, if you've got a light strand and a heavy strand, this DNA here should weigh an intermediate weight. So we would expect, again, the parental generation would be heavy. Then the very next generation, if it's semi-conservative, should all be intermediate. I shouldn't have any heavy bands left because this would have split apart. Then this will split again. The original heavy will make a new light intermediate. And then this original heavy will also be available to make an intermediate. But then the new light strand made in the first generation here will be available to combine with another light strand. So we would end up with intermediate and light bands. So here we have intermediate and light bands. This is what they saw. So this was proof that the DNA itself is replicating semi-conservatively. And when Meselson and Stahl were able to prove this, then people really dived in to figure out how to replicate the DNA. At first glance, this may look quite complicated, but it really isn't. Just follow along and I'll help you figure out what's going on here. So first of all, you can see we've got our double helix here, and we have split it open here, and we've unzipped it so that each of the original strands can form a template to add in new nucleotides. So you've got your original strand here with the new nucleotides being added, and then the original purple strand down here also with the new nucleotides being added. So let's start at the very first step, and I'll walk you through each of these things. The first thing that you're going to need to do, in a bacteria replication at least, you're going to have an enzyme called gyrase over here on the left. And this gyrase enzyme's job is to make sure that as this helix uncoils, it doesn't get too bunched up and tight over here so that the DNA snaps. You want to protect that. So this is going to protect the helix from snapping. It's going to relieve some of the tension in the helix coil as it unwinds. It's important that you know that. Then the next thing you're going to do is get another enzyme called DNA helicase. Helicase is this enzyme right here. And its job is going to be to break the hydrogen bonds. Remember that along the ladder here, the bases themselves are not actually joined to one another. Instead, they're looking more like this over here. They actually are just joined by hydrogen bonds. And the DNA helicase's job is to split those hydrogen bonds open so that we can unzip the DNA helix. Then the next thing you're going to need is these single-stranded binding proteins. That's what I've circled here in blue. These guys are just going to be attached very temporarily because if I split this open, but the hydrogen bonds are close enough, they'll just reseal themselves. It's called re-annealing. So instead, we anneal or join, A-N-N-E-A-L, we anneal the single-stranded binding proteins to the open strand to prevent it from re-annealing to itself and closing up on us. Okay? This creates what is called a replication fork, where things are going to be open right here at the center. Now keep in mind, this is a snapshot in time. We actually would have, if you can imagine this all being chopped off here, when we started adding these guys in here, the replication fork was only as big as it is here in the middle. So we've allowed replication to already begin, and we're going to continue doing it down this entire strand. So after I have relieved the tension with the gyrase, and I've broken the hydrogen bonds with the DNA helicase, and I've added the single-stranded binding proteins to prevent the strands from re-annealing to each other, next thing I'm going to want to do is to add in something called an RNA primer. You can see it over here on the right-hand side, or you can see it right here in the middle of the other strand down here at the bottom. These RNA primers are very important to be popped onto the strand because, for whatever reason, DNA nucleotides don't like to attach opposite when they're just free-floating and dangling like this. The RNA will bind much better to that opposite strand without having any other help. So this enzyme here called primase or RNA primase is going to add the RNA primers. And this box here represents the RNA primase. So you're going to add a primer to the end here, and you're going to add a primer to this end as well. This is something that a lot of people forget. They forget that on both strands, you're going to need to start with an RNA primer. You'll see we're going to focus in on RNA primers a little more when we talk about this strand down here, but let me just explain what comes next. So after you've added the RNA primer, I want to remind you what's happening on each of the prime ends. So on the 5' end of a nucleotide, if you look back at your other note, you'll see that that's where the phosphate group is sitting. Then you have the sugar, and the base is attached to the sugar at the 1' over here. And then you're going to have the 3' end of the nucleotide where the open hydroxyl group is. And I want to remind you that that open hydroxyl on the 3' end of the nucleotide is going to be available to add in the next nucleotides. You have to start here at the 3' end and move in this direction in order to get the next nucleotide tucked into that open hydroxyl. The enzyme that does the job of adding new nucleotides is the DNA polymerase number 3. And now I've written down here for you that poly 3 adds new DNA nucleotides to the strands, and it also proofreads as it goes. So basically, what it'll do is it'll look across on the other side of the strand, and let's say that this is an A on this side, well then it knows to add in a T over here. If this was a C, it would know to add in a G, and so on and so forth as you run down and through. You're going to add in opposite complementary nucleotides, and that's DNA polymerase's job. And it'll do that at the replication fork, so that as this splits open, I'm automatically adding in the new fresh DNA nucleotides. Okay? Now, this is called the leading or the continuous strand because once you put the one RNA primer down, you can just head towards the replication fork no problem. And you always have an open hydroxyl group at the end of the nucleotide that you're working on in order to add in the next one. But it's a different story for this side. Remember, we have to go in the 5' to 3' direction. But if you look at this purple strand here at the bottom, you can see this is the 5' end, which means that the 3' end is over on that side. So I would have to go in this direction off to the right on this strand if I wanted to add in any more nucleotides. So what happens is you put an RNA primer right near the replication fork, okay? And on that RNA primer, here I've written it down here, you've got the 5' end and you've got the 3' end of the primer. So you add in nucleotides in this direction. But the problem is that you need to go towards the replication fork. So what will end up happening is the RNA primase, you can see it's already been removed here. I'll tell you about that in a second. But you would have had a primer here and it would have run in that direction with the DNA polymerase 3, adding DNA nucleotides. Then you add another primer in and you run it in this direction, again, 5' to 3' direction. You're going to have a bit of a gap because the DNA polymerase 3 does not join this gap here. Then you're going to do it again, another primer, and you're constantly running in these short little sequences. That's why this is called the discontinuous strand, because it's made up of all of these little fragments called Okazaki fragments. It's one of my favorite words in all of DNA. So Okazaki fragments are going to be in this little short fragments all the way along. And it's also called the lagging strand just because it takes longer. So this one can be one primer, one and done, and you head in the correct 5' to 3' direction. And these ones will be the short sequences. It takes longer. It's more energy to try and replicate this complementary strand, the anti-parallel strand, than it is to do the actual original normal strand here up at the top. Okay? Now, I cannot leave the primers in. I do not want DNA to have some RNA and nucleotides in there, partially because they'd have the wrong bases. You'd have U's instead of T's, and you want to have thymine, not uracil. And then secondly, it's the wrong sugar. You have ribose sugar instead of deoxyribose. So the very next job will be done by something called DNA polymerase 1. Now, unfortunately, they didn't put the DNA polymerase 1 into this picture, but you can see already that the red RNA primer has been removed here, and it was done by polymerase 1. So it's going to come along, it's going to chop out the RNA primer and replace it with proper DNA nucleotides. That's going to happen here again. Chop out the RNA primer, put the right DNA nucleotides, and it's just going to go in this direction all the way along, and it's just going to be replacing RNA with DNA. It'll do the same thing once on this strand as well. Get rid of this RNA primer and put the right DNA nucleotides in there. Now, the fancy word that we use to chop out or cut out those primers is called excise. They're going to excise the RNA primers. Please begin to use the correct language as you're trying to describe this. Okay? So poly 1 cuts out the RNA primers and replaces them with DNA nucleotides. Then finally, there's a couple more things that have to happen. You're going to have to join the phosphodiester bonds that need to be formed between the sugar and the phosphate backbone over here. And that gap is going to be joined by DNA ligase. They've shown the DNA ligase here in this green little structure. Okay? So DNA ligase is going to do the job of sealing the fragments, the Okazaki fragments, to make one long continuous strand at the end of the day. Finally, you're going to have something called DNA polymerase 2, and that's going to proofread the new strands and correct any errors. DNA poly 3 has a bit of proofreading that it can do, because as it's reading the opposite nucleotide and adding things in, if it makes a mistake, it can correct it. Then as the DNA poly 1 is reading along and it's reading and looking for these RNA primers, it can be reading the DNA nucleotides as well. And if it sees a mistake, it can chop it out and fix it. And then poly 2 can do the exact same thing. It'll come along as a final check just to make sure that all of these DNA nucleotides are complementary to the strand that's opposite to it. So you actually have 3 levels worth of proofreading, which is great. You do not want your DNA to have a mistake in it, because the DNA of a mistaken DNA in a cell will be replicated over and over and over again as you make the new cells through mitosis. And you don't want all of the baby cells after that down the line, all the descendant cells, to have that same mutation or that same mistake. Because sections of DNA like this could form a gene, and the gene is supposed to be there to code for a protein. So you really, really do not want to mess with the DNA code. So it's excellent that there is triple checking. Let me just run through it one more time just so that you've got the complete story. So again, the gyrase is going to relieve the tension as the helix unwinds to prevent it from snapping. The DNA helicase is going to break the hydrogen bonds between the 2 strands to allow them to separate, to unzip. Then you're going to add the single-stranded binding proteins to anneal them to the two strands to prevent the strands from re-annealing to each other. This is going to create a replication fork. Then you're going to have the RNA primase once on the leading or continuous strand, and then many times on the lagging or discontinuous strand. The RNA primase will add RNA primers, which are starting points for the DNA nucleotides to be added. So we're going to have them always added to the 3' end. The DNA polymerase number 3 will add in DNA nucleotides starting out there and go continuously towards the replication fork on the leading strand and discontinuously towards the replication fork on the lagging or the discontinuous strand. Okay? That'll form short Okazaki fragments. You're going to need to have your DNA polymerase 1 come along and excise all the RNA primers at the front of the Okazaki fragments, and once on the leading strand, remove the RNA primer and then replace it with the proper DNA nucleotides. It's also going to proofread as it does that. And then finally, you're going to have to join the gaps in the phosphodiester bonds with the DNA ligase, and then proofread one more time on both strands with the DNA polymerase 2. Please take a look at the other handout that I've given you. It's called the DNA replication steps handout. It has all of what I've just said written out in detail. Please spend time memorizing that. You're going to be asked for details on what each of these major players does. You'll also could be asked questions about the terminology like Okazaki fragment, lagging, discontinuous, leading, continuous strands, all those kinds of words. Understanding that you're always going in the 5' to 3' direction, all that kind of stuff, very important for when I'm going to test you next. So please make sure you have all those details down. You're also going to have to answer questions. For example, if I asked you what would happen if the DNA polymerase stopped working, then you'd have to know that the job of DNA polymerase is to add the DNA nucleotides. So then you could say that you wouldn't get a new strand formed because the DNA polymerase 3's job is to add the nucleotides. And without that, no new strand. So same thing if I said, the primase is broken, what would happen? Well, if you can't add RNA primers, there's no starting point for the DNA polymerase 3. So again, you can't get the strand. So you need to know what each of their unique jobs are so you understand if they're damaged or broken, what would go wrong. All right? So make sure you spend a lot of good time on this diagram and on the written steps that are in this handout separately on a different page. And finally, I just want to mention a couple differences between how eukaryotes are going to use DNA replication and how prokaryotes are going to use DNA replication. So just a reminder, eukaryotes are animals, plants, fungi, protista, anybody in these kingdoms, whereas prokaryotes are all the bacteria, whether they're archaeobacteria or whether they're eubacteria. Okay. Now, eukaryotic genomes are massive. They're huge, huge, huge segments of DNA. So you're going to see that they don't just have one little origin of replication. You're going to see tons of them, lots of them, so that you're not just starting at one end of the DNA and hoping that you eventually get to the other by the time you need it. You just do this over and over and over again, and then these sections will eventually join to one another, and you'll get your two finished semi-conservative daughter DNA molecules. Okay. Whereas in prokaryotes, because they only have one circular DNA chromosome, and this will be the same thing that happens in their little tiny plasmids as well, you have one origin of replication, and then you're just going in both directions. And then when they meet back at the other end, you're done, and you've got your two separate identical DNA. Okay? So keep in mind, that's the difference. Eukaryotes have thousands of these little origins of replication along their massively long DNA strands that they're trying to replicate, and these guys will just have the one, and it goes in both directions, and this one goes in both directions as well. And then just finally, just zooming in on the lagging strand, just to help it make a little bit more sense. So again, you've got your RNA primer, a little section of RNA that is complementary to the original DNA strand, and you're going to have your DNA polymerase 3. Once it reads the 3' end of the RNA primer, notice here's 5' end, so this is 3' end. It's going to add in those DNA nucleotides to make that short segment, which will be the Okazaki fragment. Then you'll have the DNA polymerase 1 come through, and here it is here, and it's going to chop out that RNA and replace it with DNA nucleotides. But you've still got these gaps left. So then the gaps are then sealed by creating phosphodiester bonds between the sugar and the phosphate of the next nucleotide that's there. And that, again, is done by DNA ligase. And that, my friends, is the end of this lesson.